Hi everyone, we are Group 8 and today we will be exploring how the immune cells of our innate and adaptive immune system responds to the retrovirus known as human immunodeficiency virus, or HIV. HIV is widely recognized as a sexually transmitted infection as it is primarily transmitted through sexual intercourse. Transmission can additionally occur through cross-contamination during needle sharing and vertical transmission between mother and fetus during pregnancy. Some individuals experience severe flu-like symptoms while others remain asymptomatic. Prolonged infection and chronic inflammation can lead to the development of acquired immunodeficiency syndrome, or AIDS. So to attack incoming HIV pathogens, cells from both the innate and adaptive immune system subsequently work together to improve patients' resiliency to infection. Let's take a deep dive to uncover what these cells are, how they create a line of defense against infection, and how HIV targets the individual activity of each cell. Starting our journey in a mucosal membrane, the exchange of bodily fluids across the membrane can lead to HIV infections. In the early stage, infection is localized to cells expressing CD4 receptors, primarily in the mucosa, submucosa, and draining lymphatic tissues. Initial HIV contact with our immune cells occurs through an interaction between HIV's glycoprotein 120 to CD4 cell receptors on resident mucosal macrophages and dendritic cells, leading to membrane fusion and release of viral DNA into the host cell. These cells express cytosolic pathogen recognition receptors that are able to identify specific pathogen-associated molecular patterns exhibited by HIV, stimulating a series of signal cascades to initiate the immune response. Macrophage activation and the release of interleukin-1 is essential for the recruitment of additional immune cells towards the site of infection. However, HIV primarily targets the migratory macrophages as a mode of transportation throughout the body, allowing infection to spread and supports the formation of infectious reservoirs. In addition, HIV-infected macrophages can transmit the virus during antigen presentation to inactive CD4 plus T cells, causing increased rates of CD4 plus T cell infection and depletion. Dendritic cells are the next target of HIV infection and express a cytosolic toll-like receptor, TLR7, that is able to recognize incoming HIV viral RNA. This recognition event stimulates the maturation of dendritic cells for antigen presentation and increased production of type 1 interferons for the activation of NK cells. However, antigen presentation can be reduced upon dendritic cell infection, often resulting in stalled T cell activation and the delayed activation of the adaptive immune response. NK cells are essential for the arrest of HIV viral replication and the protective immunity response, driven by their antibody-dependent cellular cytotoxicity. NK cell activation stimulates degranulation and the release of lytic granules, inducing apoptosis in neighboring infected immune cells. Now that we've discussed some of the major innate cells, let's discuss the adaptive immune system cells, starting with T cells. The naive T cells are activated as a result of TCR recognition of an MHC class 1 or 2 peptide and a concurrent co-stimulatory signal sent by the same APC, primarily activated dendritic cells and macrophages. HIV predominantly infects and replicates inside of CD4 plus helper T cells by binding to CCR5 receptors on their surface, ultimately leading to apoptosis. Mass CD4 plus T cell depletion can lead to insufficient B cells, signaling for antibody production, and reduce activation of cytotoxic CD8 plus T cells. Active CD8 plus T cells secrete a series of inflammatory cytokines and release lytic granules, inducing apoptosis of infected cells. Despite their critical role in controlling acute infection, prolonged infection can lead to CD8 plus T cell exhaustion and chronic inflammation. Lastly, our naive B cells become activated with stimulation from cytokines and interacting with helper T cells. The naive B cell internalizes the antigen and presents the antigen in complex with MHC2 proteins on its surface, activating CD4 plus T cells. This interaction causes the T cell to send co-stimulatory signals and cytokines to the naive B cell to initiate activation and differentiation into plasmablasts. With our plasmablasts, antibody production can begin. A fraction of plasmablasts migrate to the bone marrow to further differentiate into long-lived plasma cells and have been found to generate the largest portion of HIV-specific antibodies in the body. However, HIV infection can cause significant B cell perturbation, specifically memory B cell depletion and exhaustive B cell activity. As we have learned, HIV has a detrimental effect on the immune response. HIV patients are often highly immunocompromised due to CD4 plus T lymphocyte depletion, resulting in an increased risk of opportunistic infections. In addition, HIV infection causes chronic activation of both the adaptive and innate immune systems, resulting in a low, sustained inflammatory state that persists even after antiretroviral therapy. This has led to the new categorization of HIV as an inflammatory disease as opposed to immunodeficiency, leading research to target the inflammatory response for HIV treatment. The mechanism of HIV infection and the impact on our immune systems remains a critical sector of healthcare research. By understanding how HIV attacks, we are able to propose new targets for novel therapeutics in hopes to develop highly effective treatment methods and improve the livelihood of those infected.